Welcome. I'm glad to see there are so many people attending this last day oncology session. My name is Benedicte Bakke, and I work as a senior industry specialist in D&B International Healthcare. We host this session together with Oslo Cancer Cluster. Will we see some time in the future that our children will not die of cancer? If we look into the crystal ball, will we be able to cure cancer completely, or at least see it only as a chronic disease? I'm not sure, but I'm sure that it will be very different from what we see today. Historically, we have had 3 pillars of cancer therapy, with surgery, radiation, and chemotherapy. Immunotherapy is now seen as the 4th pillar in the treatment of cancer. It represents a game changer since its introduction in the space. Patients don't just live longer, but they also have a fairly good quality of life. However, immunotherapy is still in its early days, with breaking news almost on a daily basis. FDA's very welcomed approval of Kymriah a few weeks ago, representing the first gene therapy approved in the treatment of cancer, is a very good example of this. And I don't know if we are able to foresee how cancer treatment will be in 5 to 10 years' time. We have seen an impressive development in clinical treatment, but there are also many obstacles. Low response rates, challenging immune-related side effects, and not at least high cost of treatment. And who is going to pay for the treatment? We have a number of experts here today to present and discuss recent approaches in cancer. They are all representing different angles into the treatment, and they will present how they see future cancer therapy. The session is meant to be forward-looking and to some extent controversial, since we don't know all the answer. By this, we kick off the session, Advancing Cancer Immunotherapy by Novel Partnership, Bugs and Bites. So keep listening for the next 75 minutes and enjoy the speeches. MSD Merck has one of the 2 major medicines in the field with the Keytruda. Our first speaker, Katra Damadi, works as an executive director for oncology research and evaluation lead in MSD. She has been worked in the biotech pharma industry for more than 15 years. She has been instrumental in major acquisition deals in the field. She will give an overview of the cancer immunotherapy today. Katra, we look forward to your speech. Welcome.
Thank you very much. So it's a pleasure to present MSD to you today, and particularly our thinking in terms of immunotherapy. This is our forward-looking statement. So just a couple of words about the company before I start on the science. So we operate as MSD globally, apart from US and Canada, where we're known as Merck. We have a history going back 126 years. We have 4 business units: pharma, vaccines, biologics, animal health. Our revenues in 2016 were just short of 40 billion, and we spent 10 billion on R&D expenses. We're headquartered in New Jersey and have approximately 68,000 employees. So I'd like to talk a little bit about Keytruda first. Keytruda is our foundational program in cancer. It's an anti-PD-1 inhibitor antibody. PD-1 is expressed on activated T cells, and when it interacts with its ligands, PD-L1 and PD-L2, found also on tumor cells and other cells, this leads to dampening down or inhibiting activation of T cells and generally a negative effect on tumor immunity. So we have now seen single-agent activity with Keytruda in more than 20 different tumor types, which is quite remarkable. Of course, a lot of patients still do not respond to Keytruda, don't benefit from it, and this is something we need to deal with. So some of this activity has led to significant increases in patient survival rates in key cancer area indications such as non-small cell lung cancer, melanoma, and bladder, which has been very promising. So our strategy in oncology is really to establish Keytruda as a backbone for treatment of cancer. There's a couple of areas which we've been focusing on. So it's important to identify patients most likely to benefit from Keytruda, and we have a number of predictive biomarkers in this area that I'll talk about. And this is really to avoid unnecessary tox and healthcare costs, and also improve monotherapy efficacy, so improve the number of patients that benefit through the vast combination program that we have that I'll also talk about. So these are some of the highlights of the progress made in the last 5 years. We have actually now more than 550 trials ongoing. 300 of those are collaborations. We have approval in 10 different indications, a couple of approvals of interest. A first ever cancer approval based on genetic trait. These are the microsatellite instability high tumors, patients who have high tumor mutation burden, and regardless of their tumor location. And also the only PD-1, PD-L1 to be approved in first-line lung cancer, which is now a move away from chemotherapy, which has been the standard of care for 30-odd years. So we have a very broad collaboration program. And of course, we couldn't do this without the collaboration with our partners, both coming from academia, small biotech, and large pharma. And we've taken a very empirical approach, so we're exploring a wide variety of different mechanisms of action. And we've actually seen positive results coming from a number of these areas. So, for example, in renal cell carcinoma with axitinib, a multikinase inhibitor, we've seen response rates now moving up to be more than 70% with IDO inhibitor, an IO agent, pushing response rates to about 60% in melanoma. So we're seeing positive results coming from different areas. So as I mentioned, we have a lot of activity in terms of predictive biomarkers to select patients who most likely to benefit from Keytruda. So we're taking 3 approaches. Of course, ligand expression of PD-L1, and we have the first PD-L1 companion diagnostic approved. Patients with a high tumor mutation burden, as I said, respond more favorably to Keytruda. This makes sense because they have more neoantigens and more visibility to the immune system. And we have the first approval of patients who are MSI high, regardless of their tumor location. And then we're also doing immune-related gene expression profiling, understanding whether the tumors are inflamed or not, because patients with inflamed tumors also respond better to Keytruda. So this is some of the data relating to those biomarkers. I don't know where the point. No. Anyway, on the left, this is the durability of disease, shows the durability of disease control in mismatch repair deficient tumors, and you can see nice responses, in a number of different tumor types. And on the right, you have the interferon gamma 6 gene signature score, which has come out of our genetic gene expression profiling, and patients with a high positive score have longer survival rates and respond better to Keytruda than those that have a negative score. So beyond Keytruda, this shows a snapshot of our early stage pipeline, and you can see that it's pretty immuno-oncology heavy. So we have a number of immune agonists to increase the immune response within cells, cancer cells, so targets such as STING and GITR. And then we want to inhibit molecules that actually cause immunosuppressive environments such as IL-10, LAG3, IDO, TIGIT. And then we also developing a personalized vaccine approach, which I'll talk a little bit about. The tumor microenvironment, we think, is a very important area which we are exploring, and we have a couple of programs there. One area I'd like to talk about that we've been investing in is the innate immune stimulators. So we know that tumors that do not have T cells, so-called cold tumors, respond less favorably to Keytruda than tumors that have T cells, a more inflamed profile. So we're taking a number of different approaches to convert cold tumors into hot tumors. For example, we have a collaboration with Dynavax, who have developed a TLR9 agonist. So TLRs are part of the natural innate immune response to pathogens. And we've seen some very nice results, very early data presented at ASCO with TLR9 agonists combination with Keytruda, seen 100% response rate, which is very early. It's only 7 patients, but pretty promising. We recently announced the acquisition of the company Rigontech, which they developed a very nice program targeting RIG-I. So RIG-I and STING are pathogen-associated molecular pattern receptors, and basically the part of the innate response, immune response to viruses. So they detect cytosolic DNA in case of STING and RNA in the case of RIG-I, and produce a type 1 interferon response. We can harness that activity in a tumor cell setting with synthetic agonists of RIG-I and STING. These are early clinical programs. We're very excited to see what's going to come out of those studies. And we have a number of collaborations in terms of oncolytic viruses. We've seen very promising results with TVEC, in combination with Pembro in melanoma, and now a number of other indications. And we have a collaboration with DNAtrix in glioblastoma. Finally, I'd just like to say a couple of words about our neoantigen personalized vaccine approach. We have a collaboration with Moderna, who have the messenger RNA vaccine platform. And this is really individualized therapy. So you take tumor biopsies from patients, sequence their mutanome through algorithms and various methods, identify which mutations are likely to be more antigenic, immunogenic, and which are most likely to be presented on the cell surface. These are then formulated in lipid nanoparticles, injected back into the tumor, and then we're now in very early studies, but we're hoping to see good results there. There are a number of key issues and challenges with this approach outlined here. So turnaround time, it's really scaling out rather than up. It's individualized therapy. So there are a number of challenges, but overall, these are the areas that we're now exploring. Of course, there's a number of other areas that I haven't had time to speak about, like myeloid cells, for example, another area of biology that's going to be very important in the future. So I hope that gives you a snapshot of our activities at Merck in immuno-oncology.
Thank you, Kateri. It's a very impressive development program you have.
Thank you.
So look forward to follow it further. Alligator Biosciences is a listed Swedish company focusing on immuno-oncology for immunotherapies. Their approach to cancer therapy is antibody-based, and their technology was recently validated by a major licensing deal. Our next speaker, Per Norlén, is a medical doctor. He's an associate professor and has extensive experience in drug development. Look forward. Thank you.
Thank you. Alligator Bioscience. We're developing immuno-oncology compounds, and in that field we are differentiated, developing tumor-directed immunotherapies. That basically means that we develop therapeutic antibodies that activates the relevant part of the immune system rather than general immune activation, all in order to focus the immune attack to the tumors and reduce systemic side effects. Within that field, we have a strong pipeline of 5 antibody-based products, all with the first and best-in-class potential. And we have made a major out-licensing deal with Johnson & Johnson on the first clinical product. Success factors of Alligator are twofold. We have a very strong technology platform and also a very strong research R&D environment. And that basically comes down to the fact that we entered immuno-oncology in 2008, basically 5 years before immuno-oncology got generally accepted. And due to that, we have a very competitive pipeline today. The first product entered clinical trials in 2015. At the same year, it was out-licensed to Johnson & Johnson in a major out-licensing deal. Based on the initial payments from that out-licensing and successful share issue at the IPO last year, we today have a very strong financial situation as well. Basically money in the bank to take and build the pipeline for the next 5 years. The pipeline is completely focused on tumor-directed immuno-oncology. And just to explain the concept, general immune activation is very effective, but also associated with severe immune-related side effects. We activate the immune system more selectively in the tumor area. And one specific example is we can engineer bispecific antibodies with one immune-activating entity and one tumor-binding entity. Such antibodies can locate to the tumor area to focus the immune attack. And we can also engineer those antibodies to be inactive until they bind to the tumor to further localize the immune attack. So the pipeline. 1013 clinical trials, and it's an out-licensing project, so this has been fully out-licensed to Johnson & Johnson, run by Janssen Biotech. And I'll come back to this product soon. We have also a bispecific first-in-class dual immune activator, ATOR-1015. Here it's a fully owned project, and we intend to take it further ourselves. And we'd also like to focus on another bispecific project, which is a co-development project. So basically 3 different partnership models. 1013. And first, a crash course in immuno-oncology. We have 2 parts that are important. One part telling what to attack, that's antigen-presenting cell. And the other part performing the attack, that's the T cells. Immuno-oncology is basically the interaction between those 2 cell types. If we look at this interaction in more detail, here is presented one to attack, that's a tumor antigen. Any tumor cell expressing that on the surface will be attacked by the T cells. But that signal is not enough by itself. That signal says this is something non-self. It doesn't belong here. But in order to elicit an immune attack, you also need a modulating signal saying whether it's dangerous or not dangerous. And that's all these signals. And every signal here is on the T cells, and they have the potential to be an effective immuno-oncology therapy. Just to put CD40 into context, CD40 is the only signal that specifically activates the antigen-presenting cells. To make a long story short, it was out-licensed to Johnson & Johnson 2 years ago. We have completed 1 clinical phase 1 trial. Johnson & Johnson is running a 2nd clinical phase 1 trial, and it's a very beneficial deal for Alligator. Johnson & Johnson pays for everything. We have received $50 million today, and we have a total potential of $700 million in milestones. On top of that, very significant royalties on all future sales. With that, I'd like to move on to the next asset, which is 1015. Here is a first-in-class dual immune activator. It's been specifically developed to be the perfect combination with PD-1. So PD-1, it's very good at activating T effector cells. But that also induces counter-regulatory mechanisms, suppressive mechanisms, notably regulatory T cells. And both targets, OX40 and CTLA-4, were selective because they are strong suppressors of regulatory T cells. The fact is that, though, that 1015 adds another dimension. By putting them in the same molecule, we have the opportunity of bringing cells together, creating a physical bridge between cells expressing these two receptors to increase cell-to-cell communication. And that can give additional synergy. Today we have demonstrated that synergy. Here's an example of regulatory T cells. The yellow line is the combination of OX40 and CTLA4 antibodies. And the green line above is 8 or 1015 when they're put in the same molecule. So 4 to 5-fold increase in the suppressive effects. And we have also confirmed this on the other side in tumors. So here we can demonstrate that we improve this T effector cell regulatory T cell ratio in tumors in several different models. And the effect is far superior to either antibody by itself. This compound we are developing ourselves, and we intend to bring it up to clinical phase 2 before out-licensing. So we do not plan partnership at the moment, possibly for clinical co-development, but not for out-licensing at the moment. The next product is a different structure. Here, it's a co-development program with a US company, Aptiv Therapeutics. We have built this compound together using our proprietary technology platforms. Alligator has added our human antibody library and the protein evolution technology FIND, and Aptiva added the bispecific technology Adapter. And we have generated a first-in-class product which has a tumor-localizing properties. So essentially, we will inject this product in the circulation. Once it reaches a tumor, it will stay there due to the tumor-binding entity. That's this section here. And then we can focus the immune attack to the tumors. But in addition, it's made inactive. So it's engineered to circulate throughout the system and be activated only upon binding to the tumor. All in order to focus immune activation to the tumor area as much as possible. So I'd just like to conclude by saying that we now with 1015, the first-in-class dual immune activator, and 527, a first-in-class tumor-localizing antibody, we believe we are at the very forefront of bringing immune oncology and bispecific immune oncology antibodies to the next generation. Thank you.
Thank you, Per, for a very good overview. It's very impressive to see what you have achieved so far. So we look forward to follow you in the future.
Thank you.
Roche is one of Switzerland's mastodons in pharma and share Basel as headquarters with Novartis. Our next speaker is not based in Basel, but he's based in Munich. Roche has had oncology as a long-term focus, and external partnerships are a cornerstone of their R&D strategy. Stefan Ries is Global Head of External Innovation. He has been with Roche since 2006. He's responsible for all externally derived products and assets in oncology, and he will focus on the future and look very forward if we can see if we can treat our children and not experience that they will die of cancer.
Thank you so much for the kind introduction. So what I want to do, I don't want to talk too much about the Roche pipeline. I mention sometimes our projects, but I want to give you an overview on what we think is our perspective on the future of oncology and what we think are promising approaches and therapeutic approaches that are currently being developed. And I also try to look a little bit into the future. So in principle, it's gazing more or less into the crystal ball. And I think nobody can really say 20 years ago one would have predicted what is nowadays reality, like the cancer immunotherapy. But nevertheless, I want to give it a try. So why is cancer so difficult to treat in the first place? So in principle, cancer is a genetic disease. So cancer, each cell is different. It's loaded with mutations. It's polygenetic. It's highly unstable. It makes a evolution in our body and while it arises, and that makes it very heterogeneous. So all the cancer cells within our body are different. There's differences between tumors at different sites in one patient. And there's also a difference, of course, between tumors of different patients. So it's a highly heterogeneous disease. It's not one disease, it's actually 1,000 diseases. And finally, it's also a systemic disease. So all cancer, and I think this is really important to remind ourselves, starts as a local disease. But it then spreads pretty rapidly and it becomes a systemic disease. And once it has spread, it is virtually incurable. So people die of the disease usually once it's diagnosed. So this all has changed, and I think this is really absolutely amazing and stunning news with cancer immunotherapy and with the checkpoint inhibitors. And you see here some checkpoint inhibitors that are currently in clinical trials, PD-L1, PD-1, CTLA-4 antibodies. And you also see data, actually not from Roche, but from our competitors. And we find also these data extremely exciting, that in metastatic melanoma, we have now follow-up data of more than 10 years. And you can see that this curve reaches a plateau after 10 years follow-up in overall survival. And that means we can speak for the first time of cures in these patients that would have otherwise died from their disease within a couple of months only. And if you look at the PD-1 data, and we just saw a very nice overview also from my colleague from Merck, the PD-1 data look also very promising. So after 5 years, we have now overall survival data as a follow-up. And they are even better. They have 34% overall survival. And even other tumor types like small cell lung cancer, they now start to respond to these treatments. And after 5 years, we have about 16% overall survival, which is absolutely outstanding for this type of disease. So I think this is really remarkable that for the first time, we can now speak about thinking at least about cure. So you may all know the cancer immunity when it arises in the body. It's a multiple step process, and there was a very nice drawing by Dan Chan and Melman from Roche, actually Genentech, and they basically have depicted this as a cycle, as the cancer immunity cycle. It all starts when the cancer cells die, antigen is released, this is taken up by antigen-presenting cells, usually dendritic cells. They prime T cells, naive T cells. These T cells, they traffic to the tumors, they infiltrate the tumors, they recognize the tumor cells, and finally kill the tumor cells, and then it starts all over again. So this is how a normal cancer, or every immunity actually in our body works. But because cancer is developing in these patients, something must be wrong. And if we look at these patients, there are basically different types of immune phenotypes. And different tumors have different immune phenotypes, and therefore we also expect they need different therapeutic solutions. So we have on the one extreme melanoma and lung, and I have shown you some data. And these cancers, they are actually mostly full of CD8 positive T cells. Nevertheless, they don't respond. And these T cells, they can be activated in a majority of patients at least with checkpoint inhibitors. So we are basically intervening in the later part of the cancer immunity cycle. On the other end, you have the other extreme, like ovarian cancer, gastric cancer. Those cancers are pretty much immune deserts. You don't find a single CD8 positive T cell in these cancers, not even in the patient's body, in the periphery, you find any T cells that would recognize these tumors. So these requires a completely different treatment. So we think we have to increase the number of antigen-specific T cells and basically increase the antigen presentation, and then there's everything in between. So this is like a continuum, more or less. So in the next couple of slides, I want to highlight some technologies, some approaches we find very exciting. They are somewhat looking into the future because some are approved, some are not yet approved, but some of them are already in clinical trials. And the first one are the direct T-cell engagers. So for a direct T-cell engager, you don't really care a lot about your immune system. You directly link any type of T-cell that's in your body, in the tumor or close to the tumor, with a certain construct. In this case, the first one was the BiTEs, developed by Micromet and Patrick Boyerle, to cross-link any T cell via a CD3 epsilon binding to your tumor antigen, and by that you cross-link it, the T cell kills the tumor cell, and this led to the approval of blinatumomab in B-cell ALL in 2014. I think this was really amazing what responses this drug achieved in this late-stage relapsed patients. There is one disadvantage with this approach, and this is they have a very short half-life. In these patients, they require a micropump all the time to keep the levels high enough. And therefore, there are companies now, including Roche, ourselves, we develop next-generation BiTEs or T-cell engagers. And one is shown here. This is our proprietary platform. So we call them TCB, T-cell bispecifics. They have 2 binding arms, which create avidity for the antigen, tumor antigen. They have a CD3 epsilon binder, and they have an inert engineered Fc region that allows to have almost an antibody-like half-life in patients. And what you can see here, this was presented at ASCO earlier this year. We now see for the first time with this construct really not only efficacy in hematological tumors, but also in solid tumors. And in this case, we have monotherapy, but also combination therapy efficacy in third-line colorectal cancer patients that had otherwise no treatment option any longer. So this is also a very promising approach in our eyes. Another thing that Roche is currently not actively involved are cellular therapies. So you have heard a lot on this conference. And mainly the adoptive cell transfer of T cells. There are different approaches. One is the CAR T cells and the transgenic TCRs. So this is more or less an ex vivo gene therapy. And then there are also the TIL approaches. They take the tumor infiltrating cells out of the patient's tumor. They expand then these T cells. They don't modify them, but then they infuse them back and also get dramatic responses, at least in certain type of tumors like melanoma. And you have also heard that this led to the first approval actually of the Novartis compound CTL019, which was approved for, again, relapsed and refractory B-cell ALL based on really stunning Eliana phase 2 trial results that showed an overall remission rate of this highly pretreated patients. And these are young patients really that had no other therapeutic option any longer, of 80% and more. And there's also a second drug from Kite under review currently for the same target, but for a different disease, namely for aggressive non-Hodgkin lymphoma, also under review by the FDA. So we think this is very promising. However, there are some hurdles to overcome, and that's why we have not yet really started too much to invest into this technology, and one is efficacy. So many of these exciting data are in hematological diseases, hematological cancers, and in solid cancers it's much more complicated. You have a tumor immune-suppressive tumor microenvironment. You have many things that make it harder for these T cells to work. And I think we need to have a better understanding because most of our patients suffer from solid cancers, and we need also to have a better understanding how to overcome these hurdles. Also in terms of safety, there's still some cytokine release associated with this treatment, so it's a pretty tough treatment for these patients. And we also want to increase the selectivity of these treatments for cancer. Ideally, one could think not using autologous T cells, but off-the-shelf T cells. That would be the best, not only for the company itself, of course, because it's not an individual product, but it's also for the patient better because the cells would be immediately available. These patients, they wouldn't have to wait until the T cells are taken, are modified, and are infused back, because this is all time that counts for these patients in order to survive. So we monitor this field. We have not stepped in yet, but we are very interested in this approach, and we think it's absolutely exciting what's going on in this area. Oncolytic viruses is another thing my colleague from MDS from Merck just mentioned. So initially the idea is very old. So 100 years ago, people realized when they had heavy infections, sometimes they were cured of their cancer. And this was completely independent. This was just an observation. So about 30 years ago, people tried to modify viruses in a way that they could replicate in tumor cells, but not in normal cells. And they thought these viruses will just wipe out the tumor, lyse the tumor is gone, virus is gone too, because there's no host anymore. This turned out to be a bit more complicated, but also again fits well to cancer immunotherapy, because nowadays we know when these viruses replicate in cells, they cause a local inflammation. It's like an adjuvant effect, and they release tumor antigen. And this nicely again feeds the whole cancer immunotherapy reaction. And now there are first data actually on TVEC, an oncolytic virus developed by a small biotech company, but later licensed from Amgen in melanoma again. And recently, I think 2 or 3 weeks ago, there was a really interesting paper published in Cell from Antonius Ribas that showed absolutely exciting combination data with PD-1 therapy. So there are many viruses currently being tested, and as you see, there are many oncolytic viruses in clinical trials. We think this is absolutely exciting also for the future. Personalized vaccines, I don't have to say too much because it was mentioned before, this is very personalized. Cancer vaccines in the past so far unfortunately failed, But this is a completely new approach. People realize that you really need to target neoantigens that are only present in the patient's tumor and nobody else, because that somehow makes this available to really generate T cells against these neoepitopes that would be otherwise affected by central T cell tolerance. And in this case, originally there was also a lot of skepticism, but recently there were 2 papers also in Nature that showed promising results from BioNTech, which we collaborate, actually our partners at Genentech, and from Neon Therapeutics. And we also have another interesting approach going on called Squeeze Biotech. This is actually a relatively simple technology. So in principle, you do a microfluidic squeezing. You squeeze the cells really physically. And by squeezing the cells, you don't hurt them so much that they die, but you open up certain pores. And these pores basically lead that you can fill the cells with whatever therapeutic you want. And we can use them, for example, to make B cells as therapeutic vaccines. So not dendritic cells, because they are pretty difficult to handle, but B cells. So this is currently things we are doing also in Roche. Finally, the immunosuppressive tumor microenvironment. I think this also deserves a couple of sentences. So there are now more and more data that indicate that you cannot really target a solid tumor because there's a lot of immune suppression going on in these tumors. There are tumor-associated macrophages, MDSCs, Tregs, fibroblasts, many more things. We ourselves, we have a program that is targeting cancer-associated macrophages via the CSF1 receptor antibody emactuzumab, and we also recently published first results from the clinic in Cancer Cell and in Lancet Oncology in 2015. So we think this is also a very interesting area. So overall, I focused a lot on cancer immunotherapy, but I think there's much more to come. It's almost impossible to cover in an hour, and certainly not in 15 minutes, what is cancer immunotherapy able to do. So we heard again, innate immunity may be an important field because everybody focuses on CD8 cells, on T cells, but innate immunity is always the first immunity that kicks in before the adaptive immunity kicks in. So you have to somehow make sure that you hopefully locally can activate innate immunity, toll-like receptor agonists, STING agonists, many other things that are possible. The microbiome, I didn't even mention so far because this is a complete new era within cancer immunotherapy. So there's more and more data, mainly from Lawrence Sitvogel, but also from other labs worldwide, that the microbiome in each patient basically determines whether you respond to a therapy or you don't respond to a therapy. Maybe we can use this later on for patient stratification, and in the future we may be even able to use the microbiome information to treat cancer patients by changing their microbiome. I think this is still not tomorrow, but it's at least something that people are currently working on. And then there's much more to come. 2 things at the end, because I don't want to give you the impression that now it's cancer immunotherapy and we work on cancer immunotherapy and the field is now, cancer is solved, there is no cancer anymore. That's certainly not true. I mean, cancer immunotherapy gets a lot of attention, but we shouldn't forget about the classical oncology. There are now new approaches that allow drugging the undruggable. So there are really good oncogenes known, RAS, for example, MYC, beta-catenin, nobody is able to target them. Also, we know them for, I don't know, 20 years or longer. And now there are recent data that show that via exosomes, which are an ancient system of intracellular communication, you can use exosomes to deliver payload like siRNAs or locked nucleic acids very selectively into certain target cells. And this depends on your exosome, which cells you target. And there was a good paper published recently in Nature again that shows they could use this approach to target KRAS mutant pancreatic cancer. Finally, targeted protein degradation, dicronomides. This is also something very new. So you use a small molecule that links the target with an E3 ubiquitin ligase, and by that you link the 2 molecules and you do an intracellular degradation. You completely degrade your target with a small molecule that was otherwise impossible, and that is good for targets that have no catalytic inhibition. I want to close. I know I'm over time. This is our road to future of oncology, so we are not yet there. We currently treat the metastatic disease, chemotherapy, targeted therapies, cancer immunotherapy, it's all good, but it's still metastatic disease. We need to go to the early stage disease. I think we need to get better in diagnosing cancer early, because then you can cure cancer completely by surgery. We need to get better diagnostic tools that allow routine screening and less aggressive treatment in the end. And optimally, we would at some point reach a state where we can even prevent the disease, at least for high-risk patients that had otherwise no choice. We are in the beginning for HPV-induced cancers, but maybe in the future we will also have some other cancers where this is possible. And with this, I want to end. Sorry for going a little bit over time.
You are forgiven. Thank you, Stefan, for an excellent thank you for an excellent overview of the different treatment approaches. Very interesting. Gut bacteria plays a crucial role in building up our immune system at birth. There is increasing evidence that a dysregulated gut microbiome are strongly involved in inflammatory bowel disease, metabolic diseases, and oncology. Enterome is involved in therapeutic approaches for microbiome-related diseases. Our next speaker, he's a serial entrepreneur, and this is the third company he's scientific officer. He has more than 100 scientific publications and inventor of multiple patents. So he will focus on how a microbiome is approached in the treatment of cancer. Welcome on stage.
Thank you, Benedict, and thank you, Stefan, for introducing my talk about the microbiome and how novel the microbiome field, and especially, so what I will try to explain to you today is why people believe the microbiome is important with respect to immuno-oncology. I would say it's probably important to any type of human disease. And second, how we, at least at Enterome, we are trying to use the microbiome to potentially identify new targets and new drugs that maybe one day will fulfill your dream at first. So let's see. 2 words about Enterome. So we are French, Paris-based biotech. I'm not French, but same accent on my side. And so 2 words about the microbiome, so that everybody has an idea of what we are talking about, and especially why the microbiome started to generate such an important, significant interest, if not excitement. I think the major point here with the microbiome, which means all bacteria all of us have in our guts here, the huge interest came from the discovery that we have all of us more than 500,000 genes, new genes lying in our guts here, all together sequencing thousands of different individuals. There are more than 10 million genes, and when I mean genes, essentially new genes. We put a cutoff at in between 70 and 80% identity, so it's a huge amount of genes. So everybody's pretty excited about that. I see it as kind of a concern in the sense that it's already difficult to deal with our own 25,000 genes. How are we going to deal and manage these 10 million genes here? And this is why what I'm trying to show you later. Just before that, all of these bacteria produced an estimated 3 to 10,000 different metabolites. You don't know, then 60 to 70% of all of our circulating metabolites in our bloodstream come straight from the microbiome. What we have identified in our 10 million plus gene database at Enterome is more in between 1 and 2 million proteins, enzymes, small peptides, highly constrained, blah, blah, which are secreted. And if they are secreted, they may impact potentially on different bacteria and regulate their growth, but maybe more importantly, they may impact on gut lining receptors, probably GPCRs, ion channels, and new things. So we believe there is a full work to do here to rebuild and reconstruct this, we call it metasecretome in vitro, and test it for activity. And I will allude a bit how we approach that. And the third class of compounds, so the first class is small metabolite, peptides, proteins, enzymes. The third class, which I believe is very important, is epitope mimics. And essentially, one of the main questions we asked at Enterome is, if you take a 9 amino acid epitope for human autoimmune disease or involved in immuno-oncology, this 9 amino acid epitope is going to be unique on a 25,000 genes genome. However, if you look now at 10 million genes, this is not unique. The vast majority, if not all epitopes we have a very good and decent epitope mimic in the microbiome. And to what extent this can generate some autoimmunity, autoimmune reactions against self in oncology, in diabetes, in MS or others, is probably worth investigating. But anyway, these are the three classes of factors we are interested in at Interhome. And jumping to immuno-oncology here, I think there is essentially no point that the microbiome does impact on checkpoint inhibitors response. I think the major questions are first, the level of the impact, to what extent is it really significant, yes or no? And second, and maybe the first one might be only answered after the second will be answered, what are the true effectors mediating impact on checkpoints? And what is absolutely clear, and I refer you to all of these papers here, what is absolutely clear, at least in animal models, is the microbiome and its composition affects the response, but also the toxic side effects linked to checkpoint inhibition therapies. And there are a couple of nice papers. You mentioned Sid Vogel before. There are others as well. And I'll refer you to this paper. But I think there is no point about that. There is an impact. So what I'm going to present to you now is how are we trying to address and identify which molecular entities are driving the response to checkpoint inhibitions. And I called it, managing complexity, because the major problems we are facing is this huge diversity of genes. So we need somehow to restrict this huge diversity to something we can manage. So the principle is pretty straightforward, actually, and we call it stat to drug here. The idea is always the same. We start with very large cohorts of patients. When I mean very large, a couple of hundreds to thousands of patients. We take feces samples from these patients. We extract DNA, and here one thing which is very important, we don't perform 16S rDNA like many people. We perform shotgun sequencing, which allow us to identify genes and other things that might be important. And once we went through all of this, what we try to do is to compare healthy individuals, of course, against treated or against pathological people, and we try to identify a couple of bacteria that seems to be moving up and down according to the whole stuff. And this is absolutely key because we did it for IBD, we did it for NASH, we did it for many different things. At the end, what we find is not 500,000 different bacteria, it's only a couple of dozen of bacteria that seems to be clearly associated with an effect. And then, in a second step, we can use this knowledge of this couple of dozen of bacteria to start screening libraries that we generated from the whole microbiome. And at Interhome, we have two types of libraries. We have full metagenomics, full, I mean, it's always partial, but we have more than 200,000 clones covering more than 4,000 to 10,000 different species. The idea here is that we clone large pieces of a DNA, 50 kb. Why 50 kb? Because it should code for full metabolic pathway, so you have a chance to generate a metabolite. And then, with the knowledge of which one is what, we can start digging and identify the relevant bacteria, produce whatever they secrete, and go through functional assays. So back to immuno-oncology again. As I said, the most important first step of the whole story is patients, and we need large cohorts. And you don't know, but we signed a big alliance with BMS last year. And thanks to this alliance here, we will have access to more than 1,000 different patients, all characterized, treated or not with their checkpoint inhibitors. We will take fecal samples at baseline, a couple of sampling in between, and we will check everything we see moving here during sample collection here with the clinical outcome here. And the idea will be, again, as I described before, can we identify a couple of dozens of bacteria that seems to be causally, that would be ideal, linked in between response and toxicity. Okay? And again, without that kind of database here, just forget even trying to do it. It's useless. Okay, so already short?
2 minutes.
2 minutes, okay. So just to define the two types of things that we are going to do with BMS here. 2 programs. The first one will be really biomarker studies, companion diagnostic. Can we predict before treatment who is going to probably respond, who is going to probably generate side effects, and so on. Again, I think if we don't succeed with this huge cohorts, I would say nobody is going to succeed. And second, thanks to the STAT-to-drug approach I described before, to use this knowledge to identify the small chemical entities, peptides, GPCR ligands, whatever, that may mediate a response here. And all of that, not to forget the epitope mimics I mentioned before. So that's how it works, and I don't need, because of time, to go too much into details, but we have a very robust high throughput screening. The key part here in the screening, so we have libraries, we grow individual clones, we incubate the supernatant with cell lines or cells, gut cells here. I think the assay, as you understand, is absolutely key. We need to figure out what is the right type of assay that can be predicted of a clinical response. And again, really to summarize what we are trying to do, you have the bacteria, you have the gut epitheliums here, you have receptors, and we are trying to see whatever activate, agonize, antagonize here, and what will be the output on this side here. This is why we are really focusing on secretions, small entities, and peptides. This is how a typical screen works. Usually, when we run a campaign like that, 20,000 clones, we have a couple of dozen of hits. We can purify them, go into animal models, and 2 of these compounds are entering clinic, one for phase 2, one for phase 1. Thank you.
Thank you, Christoph. Very interesting approach, and I look forward to follow your company further.
Let's see.
Companion diagnostics are an important tool to determine a drug's applicability to a specific person. Our next speaker has been in the forefront of companion diagnostics for over 10 years. He has been instrumental in Dacco's most successful product, the Hercept test assay, was involved in Agilent's acquisition of Dacco, and responsible for the diagnostic product release for the well-known immunotherapy drugs Keytruda and Opdivo. He's now working as a senior vice president at Precision Diagnostics at Immunovia. Welcome, the stage is yours.
Thank you. Thank you very much for the nice introduction, and thanks for some brave organizers to actually also invite a diagnostic company to present. Very much appreciated. So I'm not going to talk about immuno-oncology drugs. I'm going to talk about those tests that are so important to actually identify patients that are going to be treated with these drugs. So where are we at with immuno-oncology drugs and companion diagnostics? Well, on the drug side, we certainly have had some really good improvements and developments. We have, developed, several drugs within immuno-oncology. Some examples here: Keytruda, Opdivo, Tecentriq, and so forth. Really, really strong, important drugs that I would call golden bullets. Not golden bullets in a sense that they're actually going to cure cancer, but in a sense that those drugs can actually be used on a lot of different cancer types. And also, combining the treatments has, become, very, very efficient. So lots of innovative, new drugs. We also have a really good understanding of what is going on in a tumor cell, and we do know that even if we have some drugs, targeting certain pathways, we know that the tumor cells will find alternative pathways and parallel pathways, because in the end, the goal of the tumor cell is really to proliferate and survive. So we have all this fantastic new knowledge, good drugs, and then on the testing side, On the testing side, we still stick to an old-fashioned technology known as immunohistochemistry. It's a whole 100 years old technology. It's single plexing. So even if we have a lot of biomarkers involved, we stick to one single biomarker. I think I can allow myself to actually perhaps discuss or criticize a little bit IHC, because I was heavily involved in actually both the companion diagnostic for Keytruda and also for Opdivo. But I think using this technology is single plexing. It's qualitative. It's manual and subjective test reading. It's an invasive biopsy, and actually it can only detect the later stages of cancer. So how come we actually stick with this old-fashioned technology when we are so innovative over here on the drug side and understanding how a tumor cell works? Well, I guess if you look at the requirements to a diagnostic company, a companion diagnostic company, then the requirements really are we have to develop a technology that fits right into the routine setting. We have to actually have a system solution ready that fits into a workflow. So that's very important. Also have to be able to register those companion diagnostics globally. So we need to have the power in-house to be able to register those products. There are no differences in different regions. Have to be able to produce according to what I say here, pharma optimistic forecasting, meaning that of course we don't want to end up in a back order situation with a companion diagnostics that is actually enabling a drug treatment. So certainly have to be able to produce according to those sometimes optimistic forecasting. And then finally we have to be able to follow the drug. So meaning have to be able to commercialize in every part of the world. So I guess that is probably a really, really good explanation why we are still stuck with some of these older technologies. So what I looked into here was, what are the technologies today used as companion diagnostic? And you can see, IHC, in situ hybridization, PCR, really, the technologies being used today by pharma to identify patients relevant to a certain drug treatment. So you can see, IHC back in the Herceptin days, in 1998, the first companion diagnostic. And so we focus around the HER family, EGFR and HER2 especially. And companion diagnostics were within that field. And then, ISH came along. PCR has become very, very strong, no doubt. And you can see totally, actually, it's probably the most popular test. And again, not the least because PCR is present in every laboratory out there. If you look at the biomarkers being pursued, it's still only one biomarker, or sometimes two. Here I'm not taking into account. I certainly know that some of the later tests are looking at many mutations. So for example, EGFR have some tests that are looking into 50 different mutations, but in the end, it's EGFR. So that's why biomarker numbers is really perhaps up to 2. Test material still very invasive, still formalin fixed paraffin embedded tissue. Still the old-fashioned way of doing it. But looking at this table and looking at the years gone by, you will actually also be able to see there's a clear trend. Trend being that we are seeing more CDX tests, no doubt. We are also seeing that it's become more genomics based. So there's a trend right now. You want to look at sequencing and genomics, no doubt. Also a trend towards a little bit more biomarkers at least. You can see now it's 1, 2, 1, 2 in the later years. Liquid biopsies is actually now being much more popular and again really good also for the patients. And also we'll see more and more drugs actually in the label of these companion diagnostics. So that's the trend we are looking at. And these trends all come from customer requests. But important is to understand when it comes to a companion diagnostic, there are actually 3 customer segments which you have to fit into, your design and development. You have the pharma partners, of course. They are the ones developing the drug, and by that kind of, specifying the companion diagnostic. But you also have the routine laboratories. That is where the test is going to be run. And they are very much focused on a workflow approach, a full system solution. Even, it needs to be, easy to adapt into the laboratory, also space-wise. And then you have all the Googling patients. Those are the patients that are looking to be tested. So you have to kind of fit all these, customer requirements into your design and development of a companion diagnostic. So that's why, if you look at, the scheme I put up here, so customer requirements according to different parameters, and then you can see how are we doing today on companion diagnostics. So customer requirements, multiplexing, combination treatments, broader intended uses of these, tests, liquid biopsies, high analytical robustness, easy market access, and it needs to be reimbursable. And then you can see, for IHCs and PCR, if it's red up there, there are, certainly some challenges. If it's yellow, we are moving in the right direction. And if it's green, we're kind of okay. So plexing-wise, there's no doubt IHC and EC is heavily challenged. And even for NGS, which is an upcoming technology, the analytical robustness is a challenge. It's still not robust and reliable out there, in certain or in some laboratories, yes, but not in all laboratories. And there's also something about the market adaptation. So we know kind of the customer requests. I think there's one unspoken request which is very, very important, and that is actually the early detection. I think if you look at this cartoon here illustrating how a cancer typically develops from normal cells into full-blown cancer over here. I think what is very important to realize is that over here on the full-blown tumor, you have many different cell types. It's really heterogeneous. And pharma today is really trying to treat those ones over here. It's really difficult because you are targeting different cell populations, and therefore you have relapses because the other cell types are suddenly being the ones that are driving the tumor. So I think actually realizing that if you can detect cancer earlier on, pharma will be actually able to treat tumor cells that are more homogeneous, and thereby a higher success rate of their drug development. So diagnosing, old-fashioned technologies are only able to diagnose very late stages. Even tumor cell DNA and NGS is looking at DNA circulating in the blood, and that means the tumor has to be established before they can actually identify the tumor. Whereas if you take a more systemic protein response and look at the immunoregulatory response in the blood, you will be able to detect it very, very early on. And if you do that, for example, with a technology like the IMRAY technology, you will be able to actually identify it and treat it much earlier on, and by that, a higher success rate. So, last slide is really, tomorrow's CDX test. Plexing-wise, it's going to be multiplexing. It's going to be, predictive signatures we need. Clinical sensitivity, early detection of the disease is very important. Clinical utility, multiple, drugs into a CDX label. We even, see the universal CDX, perhaps coming. Testing material, liquid biopsies, needs to be safe and effective. Moving towards point-of-care testing with these technologies. And then, of course, probably have to discuss also with pharma when it comes to the reimbursement. Should it all be on the drug or perhaps also some on the testing side? So, thank you very much.
Thank you for a very interesting presentation about diagnostic test, and it's for sure it's an important tool in future cancer therapy and early diagnosis. Artificial intelligence and machine learning is all over us. We know that it will impact our lives tremendously, and to what extent we do not know at the moment. But we do know that machine learning is a powerful tool for exploring biological data, and it can be used for effective patient selection. Our next speaker, Richard Stratford, is CEO and co-founder of OncoImmunity, which is a member of Oslo Cancer Cluster. Richard, we look forward to hearing you.
Thank you for the introduction, and thank you to the organizers for giving me an opportunity to present OncoImmunity. Okay. So OncoImmunity is a Norwegian bioinformatics company. We're based at the Oslo incubator, which you can see on the slide here. It's a really nice milieu of small and medium-sized companies all focused on finding new cures for cancer. We're also next door to the Radium Hospital and also the Institute for Cancer Research, so a really exciting milieu to be involved with. So our company is a software development company. We develop machine learning-based software based to basically empower personalized cancer immunotherapy. And our current focus is really on finding the gaps in terms of being able to make accurate and good neoantigen predictions. And I'll explain a little bit more about what a neoantigen is shortly. So what is machine learning? Well, to quote one of the pioneers of the field, machine learning is a part of computer science that allows computers the ability to learn without being explicitly programmed. And machine learning is used to make predictions, it's used to classify objects, and it's used in pattern recognition. And actually, it's really beginning to permeate everyday life, and it's been applied to things like as simple as spell checkers to self-driving cars. But we are not applying it to self-driving cars. We're very much focused on applying machine learning to solving problems in immune oncology, and particularly neoantigen prediction. So what is a neoantigen? Neoantigens are created by mutations in a tumor which change the amino acid structure of a protein. And they change it to a sufficient extent that that protein then looks foreign to a patient's immune system. And the immune system can then be coaxed into responding against that target and actually destroy tumor cells whilst leaving healthy tissue unharmed. So how are neoantigens identified today? Well, typically you take material from a tumor biopsy, you isolate the DNA, and then you use next-generation sequencing to identify the mutations. But then the trick is to find out which of those mutations actually have the potential to be immunogenic, and that's where our software comes in. So what our software does is it interrogates each of the mutations and the associated protein peptides, and it basically interrogates them using a series of advanced machine learning algorithms that determine whether that peptide has the right features to be immunogenic. And the output of the software can be used to inform the design of personalized vaccines, personalized T-cell therapies, and also they can be used as generic biomarkers for patient stratification. Okay, so what our machine learning approach really does is model the antigen processing pathway inside the tumor. I can't work this. Yeah, nope, I'll give up. So the gray circle here is the tumor, and the pathway you see here is the antigen processing pathway within the tumor. And what this does is determine which peptides or mutated peptides can actually be transported to the surface of the cell and have an opportunity to interact with the T cell. So you can think of this pathway as a series of checkpoints which determine which peptides or mutated peptides can actually interact with the T cell. So what our software does is actually model each one of these checkpoints and evaluates the peptide to see does it have the right features to successfully navigate that checkpoint. Yep, so how do we develop our solutions? So let's focus on the step here, HLA binding, number 4 in the diagram. So this is how we actually develop a machine learning algorithm. So what we do to start with is compile a large data set. So machine learning is completely dependent on having good data sets, large data sets, well-structured data sets. So in this case, we've taken peptides that we know can bind to a given HLA allele, O2, O1 in this case, and we've built up a whole data set ranging from peptides we know bind really well, to peptides that bind okay, to peptides that don't bind at all. And this information has been identified using in vitro binding assays. So we build up a data set of peptides with their associated binding affinity. We then vectorize that information and use it to train a machine learning algorithm. So what the algorithm does is try and basically understand the mapping between the peptide sequence and the affinity and model that. So once we actually have an algorithm which we think is working well, we test it on de novo data. This data is data which we have known binding affinities for, so we can actually check the predictions against the ground truth and see how well we're performing. So a typical example here is shown in the diagram on the right-hand side here. So here, as you can see, most of the data points map to the diagonal line here, which shows that most of the predictions marry very well with actually the true values. And if you look at this, the gross metrics for how well this is performing, we have an error under the curve evaluation of 0.95, which is very good, and a Pearson's coefficient of 0.82, which again is very good. Now, actually, in the human population, there isn't just one or two HLA molecules, there's actually thousands of them. So we've also developed PAN solutions that allow us to do similar types of prediction, regardless of the HLA background of the patient. Okay, so this is what our pipeline looks like to date. It's focused very much on class 1 neoantigens. So the input for our software is a VCF file, which comes off the sequencing machine. So this is a list of mutations with their coordinates onto the genome. So what our software does is in silico translates those into peptides, and then it makes predictions for each peptide relating to how well it's expressed, how well it binds to a given HLA molecule, and how well it's going to be processed. We then feed that into another machine learning layer, which integrates that information. So this layer has been trained to learn the relationships between processing, HLA binding, and expression, which enable a peptide to successfully be at the surface of the cell. And actually, this integration step is really key to getting good results, and is working very well. So once we've made accurate predictions about which peptides are on the surface of the cell, we then have to evaluate how different they look, not only from their wild-type counterparts, but also from other self-peptides in the immunopeptidome. And then we basically rank those, and the output can then be used to design vaccines, T cells, and so forth. Okay, so I think I showed you previously how we test an individual module for machine learning by having an independent test set and see how well it performs against ground truth. But how do we actually test whether the whole pipeline per se is actually delivering something of clinical value? Well, the only way we can really do that is to test it on a retrospective clinical data. So this is one such clinical trial that was run in the US. It's quite a small trial. It was run in patients with metastatic melanoma. And in this trial, these patients were treated with adoptive T-cell transfer approach. And 3 of those patients went into complete remission. So the scientists who ran that trial, they ended up basically exploring which neoantigens the patient's immune system has responded to. So they actually developed all the peptides covering the mutations for these patients, and then they took blood from the patients, isolated the T cells, and did very complex, time-consuming T cell assays to find out which neoantigens their T cells were responding to. That's the best way to look for neoantigens, but it's very costly, very time-consuming, and it's not scalable. So we took the same inputs and we ran it through our pipeline. So what we saw was that, sorry, let me just go back. So in the assays where they did the physical wet lab science, out of the 149 peptides they tested, 7 of those were shown to be immunogenic, so less than 5%. So we ran the same peptides through our pipeline. What we found was 8 good candidates came out of that, of which 5 overlapped with the 7 identified in the wet lab study. So this gives us an accuracy of around 97% because we've been good at removing the false negatives, and it gives us a hit rate of around 62%. And that's really interesting information if you're developing a vaccine. So in this case, if we'd put 10 targets into a vaccine, you would have expected 6 of them to give an immune response. We've also applied the approach to patient stratification. So these are patients with metastatic melanoma that have been treated with IPI. In this study, they published actually the overall survival for the patients and their mutational profiles. So we've put this information through our pipeline, and then we separated or stratified the patients according to whether they had at least one predicted neoantigen or no predicted neoantigens. And as you can see, we get a nice separation of the curves here, where the patients with one neoantigen actually respond much better to therapy than those that have no predicted neoantigens. Right, so I will wrap up, and I'll just say to conclude, machine learning is a powerful tool that can be used to really help cancer immunotherapy. At OncoImmunity, we have developed a pipeline that can reliably and accurately predict immunogenic neoantigens. And we think actually solutions like ours are going to be key for empowering personalized approaches, because basically you have to develop solutions in a clinically actionable time frame. And right now we're looking for partners to evaluate our technology, to refine it and develop further proof of concept data. So if anybody is interested in learning more about the company and our technology, please don't hesitate to contact me on the information here. Okay, thank you.
Many thanks. Many thanks for a very interesting presentation combining two different fields. I really need for future therapy approaches. Gary Scarr is Vice President of Innovation for the Dana-Farber Cancer Institute. He leads the business development activities, is responsible for new company creation and management of 26 Dana-Farber spin-outs, and manages Dana-Farber's technology transfer office. He will focus his speech on Academic Medical Center as partner for immunotherapy innovation. Gary, welcome. The floor is yours.
Thank you. Well, good afternoon, everybody. I know that I am in the unique position between my talk and lunch. So what I wanted to do was give you an overview of how we collaborate, as well as combining an introduction to the Dana-Farber and its capacities and resources. Briefly about the Dana-Farber, founded in 1947, we're a leading research institute focused in oncology, and what you'll see here is that Farber's unique in that 50% of all of our activities are preclinical and research in nature, whereby the other 50% is patient care and clinical. There aren't very many institutions that are like that. One of the things that makes that happen is that we have about 200 physician clinicians or clinical research scientists. So these individuals usually have MD, PhDs, have their own laboratories, and see patients. As a result, we become a very translative. Our scientists are always looking at how the work in their laboratory can be translated to the clinic. And then from a deal-making perspective, the way I run my office is, who can we partner with that will help us get drugs to patients? To me, that's the most important factor, more so than the economics. I forgot it. So within the Dana-Farber, we have many different centers that are specifically designed to collaborate with industry. If you look at the top right corner, you can see all the different centers that we have. And these centers are designed in such a way that there are no postdocs or graduate students that work in these centers. They're all run by and staffed by industry-trained scientists. That gets us over some of the hurdles that we have from time to time around collaborating with others and dealing with issues such as publication. This is one of our newest buildings. We're located in Boston in the Longwood Medical Area. Within the Longwood Medical Area, you have Harvard Medical School, the Dana-Farber, Beth Israel Deaconess Hospital, Children's Hospital, and the partner system, which is Brigham and Women's Hospital and Massachusetts General Hospital. You can see, as an institute, we see over 16,000 patients a year. And what we do is almost every patient that comes into the Dana-Farber has their tumors sequenced. And it's a program that we call Profile. And then a little bit on intellectual property. Between 2013 and 2016, we received over 500 invention disclosures. And during that time period, almost 2,000 patent applications from, U.S. applications, provisional applications, and foreign applications. Which is very interesting data point is that over 25% of all of our new inventions come from our chemists. And I'll speak a little bit about that more in a second. And a little bit more about the institute 350 million per year in research. And you can see some of the numbers in terms of research grants, fellows, number of principal investigators. So there's over 200 principal investigators, scientists, and 400 clinicians. This is just some information on licensing between 2013 and 16. You can see the numbers, 181 licenses. Licensing income over that period of time, $113 million. And then my office also negotiates research collaborations or sponsored research agreements. And during the same time, you can see almost $125 million in sponsored research. This is a slide just to give you an idea of who we collaborate with. And not only who we collaborate with, but this slide in particular lists our startups. So we've been privileged to launch almost 30 startups over the last 10 or so years. Out of those 30, only 1 or 2 have failed. Almost all of them are alive and well, or they've had successful exits. The next slide will give you a little bit more information on the successful exits. But you know, these are just the logos of our 25 active startups, which is interesting. Checkpoint Therapeutics and Aileron, both on the left-hand side, they both went IPO in June. And Checkpoint, we did that deal about 2 or 3 years ago, and that's centered around the advancement of a therapeutic antibody. Aileron's a staple peptide company that was organized closer to 7 or 8 years ago. And then as far as successful exits, these are 4 fairly recent exits. Tensho was acquired by Roche, and you can see some of the others. Our most recent exit was Acetylon, and Acetylon was acquired by Celgene. One of my colleagues mentioned C4 Therapeutics. It's a Dana-Farber spin-out. Roche has partnered with C4. This is a very interesting startup in that, as you can see, it went Series A for $74 million, and it's a protein degradation company. So one of the great things about working at an institute like the Dana-Farber is that we're on the cutting edge of the newest science. And you can see that. This is just, you know, here's a slide about recent approved diagnostics, just being sensitive to time. And then we have a fairly large office. There's about 20 people in my office, and this is the team. And then I know I went through this really quickly, but I wanted to leave with my contact information. And if anybody wants to speak to me, you know, after we're done, I'm happy to. And thank you for the opportunity to speak with you all.
Thank you, Gary. Very interesting, and all your activities and your successful spin-outs and collaboration. Thank you. So the answer to my question raised earlier today, will we sometime in the future see that our children will not die of cancer? I'm not sure, but I'm sure that it will be very different from what we see today. We have in this session have had a very good presentation with a good overview of how the status for immunotherapy is today. We have looked into the crystal ball to see the different approaches. We have had the machine learning and see how it will impact the patient stratification and how it can be used on the biological data. So with this, I thank you very much for attending this session. Thank you, and safe travel back home.